Okay, this is the biotechnology lecture for Monday, February, let's see, today's the 3rd, so that would be the 6th. Monday, February 6th. We are starting in Chapter 4. We've covered a little bit of Chapter 4, and the material that I'm going through now will be the start of the second exam. So we're talking about protein structures. One of the things that you can do with protein structures using biotech methods is you can engineer the protein so it has different and enhanced characteristics over against its normal characteristics. So some protein engineering is very specific. So you can swap out specific amino acids by providing different codons in the genetic code, and that is called directed molecular evolution. So it's very precise. You know exactly what codons you need to change in exactly what places, and you do that, and then you test the protein for increases in activity or specificity or something of that nature. You can also introduce random mutations and then get a lot of different mutations, clone those different mutations into a cell, produce the protein, and then screen out the proteins that are best suited based on what is called speeded evolution or error-prone PCR. Once you do random mutation, then you've got a multiplicity of clones. You don't have a single directed clone, but you have a multiplicity of clones that need to be expressed, and then you'll have a variety of proteins that you can test all together to see which one has the best activity. So you have a starter group. This is wild type. Then you induce mutations in filter 2, and so these are mutated from the wild type. In filter 3, then you screen them for the ability to bind substrate and convert substrate, and then you pick the clones that do the best, perform the best, and then you have a pool of screen survivor cells. And typically what you do then is you take this round of survivor cells, you randomly mutate the code again, starting with the survivor cells. So you do multiple rounds of random mutation, and then you get increased selectivity, increased specificity. And so this is just the process of evolution in a laboratory. One example using biotechnology, some individuals have formed synthetic infectious protein particles called prions, and they've used this to study prion disease. And prions are proteins that have been folded incorrectly, improperly, and they confer their characteristics to other proteins. They actually recruit other proteins by transmitting that misfolded state. Okay, so the prions, which attract normal cell proteins, then induce changes in the normal cell proteins, and then this protein is accumulated. It's useless. Usually it's worse than useless because it will start to attack human tissues. Okay, so you have a normal protein. We call this a normal prion. It's got a normal conformation. It converts in a portion of its structure, then in red becomes abnormal. The protein is now non-functional. It's not working, and this prion can recruit other proteins to become prionic as well. Now, proteins are very fragile, so it's producing proteins is a long and painstaking process. The expression is usually the easy part. You know, you clone the gene into an expression host. You then cultivate the host cell, and the protein is produced under specific conditions. The downstream processing is usually the more onerous part. It's usually where the majority of expense is in biotechnology. So you need to purify the protein, verify that the protein is functional, and then preserve the protein so it will remain functional over a long period of time. Okay, so just to start with upstream processing, first you select a cell that can be used as the protein source. The most common cells that are selected are E. coli, and a yeast called Pichia pastoris. E. coli is advantageous because it's very easy to transform and it's very easy to grow. However, in a lot of instances, it's not very good at correctly folding protein. So you may want to consider a yeast like Pichia pastoris that has an endomembrane system that can fold proteins. Okay, so bacteria are easy. They are the fermentation processes are well understood. It doesn't take a lot of time to get a large amount of cells. You can very easily genetically modify them, just simple techniques that you could probably do in your own garage. And the expression is episomal, it's plasmid-based, so you can actually increase the level of protein production just by increasing the number of transformed plasmids in the cell. You use a strong transcriptional promoter. You want to make sure that the promoter is prolific so you get lots of copies of mRNA that will subsequently then be transformed to protein. Okay. Most common is E. coli. And E. coli, the majority of the proteins produced by E. coli are intracellular. So these accumulate in foreign bodies called inclusion bodies, and the inclusion bodies have to be purified out. You actually break open the E. coli, release the inclusion bodies, and then correctly fold and purify the protein. Sometimes you can form a fusion protein, and that fusion will be easily purified. The fusion protein usually has some type of substrate that it will bind, and that fusion then can be purified out on a column, an affinity chromatography column, and it's very easy just to take the lysate from the cells, have the fusion protein bind the substrate on the column, and then you can elute out the purified protein. Now, you have to remember prokaryotic cells cannot carry out post-translational modifications like glycosylation. But still, there are some advantages to E. coli. The genetics are well understood. You can make an unlimited amount of protein. It's very easy to grow. Fermentation is well understood. But foreign proteins will form inclusion bodies. The proteins cannot be folded in ways that would be active in humans, and so that's a big concern. So when you pick E. coli, then you have to pick a special type of protein that does not have glycosylation, that is easily folded using chemical means. And genetically engineered bacteria can be grown in fermenters or bioreactors. Fermenters are generally anaerobic, bioreactors are aerobic. And then you have computational process control that monitors the air, keeps the temperature and oxygen levels optimal for growth. And then once you have the highest amount of cells, the phase of growth that is highest, then you stimulate your bacterial promoter to produce lots and lots of protein. There's no need to have a bacterial promoter that is on all the time or constitutive. All you need to do is stimulate that promoter when the cell growth is the highest, and that will maximize the amount of protein. And here is a fermenter. It shows an air sparger at the bottom, and you've got an impeller in order to mix the culture. Foam breaker at the top that will break up any type of foaming that happens at the air-liquid interface. Then you have baffles that will prevent the formation of dead zones. These are on the sides of the reactor. Okay, and here's a real live reactor. This is looks like a bacteria called Serratia marcescens, which grows red. Sometimes you'll use a fungal transformation host rather than bacterial. And a lot is known about yeast fermentation because of experiences using yeast fermentation for animal feed, for malting of grain, and as well for beer production. Okay. And you're not limited to Pichia pastoris. There are many different fungi that can be used for genetically engineered hosts. Fungi are eukaryotic. They have organelles, and they're capable of folding the proteins correctly and also other post-translational modifications. And here are some examples of Aspergillus fungi. This is a mold that makes these different types of proteins. You can use genetically modified plants. This is my area of expertise. And instead of growing the organism in bioreactors, you can just grow them in greenhouses. Okay? And you look at a process that encourages rapid growth and reproductive rates of plants. The tobacco is a great choice, not because it's especially simple, but just because there's so much research that the tobacco industry did on the tobacco genome that we know all the chromosomes, we know its characteristics, and so it's a great choice because we have so much knowledge on tobacco. Okay. And then once the genetic material is integrated, then you can generate millions and millions of plants, grind up the plant tissues, and purify out the protein. Okay. Now, not all proteins can be expressed in plants. Plants have cellulose in their cell wall that is very difficult to break down. That makes purification difficult. And then they also have glycosylation patterns that are different from humans, and they could raise up, for a protein that's administered a lot, like insulin for a diabetic, this could raise up an immune response because there are two sugars, fucose and xylose, that are non-human that are in plant glycosylation patterns. The system that is closest to human cells would be some type of mammalian cell culture. The nutritional requirements are complex and the nutrients are very expensive. Mammalian cells grow much slower than bacterial cells or even plant cells. They're very easy to contaminate, so you need to use a sterile field, a sterile flow hood, in some cases, entire sterile rooms in order to culture the mammalian cells. But this is the best choice for proteins destined to be used in humans. Okay. You can also use whole transgenic animals, and the animal bioreactor production system is used. This is used extensively for monoclonal antibody production, especially in mice and rabbits. The mice are injected with some type of antigen, and then the mouse secretes the desired antibody, and the antibody is purified. The antibodies are then used therapeutically. They neutralize the antigen, and they protect the organism. And this is a way that we can purify out antibodies that will directly fight infectious disease. When you are dealing with transgenic animals, with mammals, like cows, sheep, goats, pigs, rabbits, mice, you target the expression of the protein to the milk, and in order to harvest the protein, you just simply milk the animal. For avian species, chickens, quail, things of that nature, you target the expression to the eggs. So you basically take the eggs after they're laid, you open up the eggs and purify out the protein from the insides of the eggs. The first transgenic animal drug that's in the market is called atrin. It's produced by genetically modified goats, and atrin treats a clotting disorder. And there is genetically modified animals. They have been produced, cows, sheep, goats, pigs. And remember, genetic modification is different than cloning. Cloning is when you have an animal that does produce a particular gene, then you want to make copies of that animal. That is cloning. But the initial genetic engineering, where you actually take the human DNA and integrate it into the goat DNA in this particular case, then you implant the DNA into an egg, a fertilized egg, and then the egg is implanted in the female. Then you test the offspring to see which ones are genetically modified, and the ones that are genetically modified, the females, then you milk them, and you simply extract the protein from the milk. And each goat can, the antithrombin or atrin was used to be harvested from human blood donors. And annually now a single goat can produce as much atrin as you would find in 90,000 human blood donations. So the prolific nature of this makes it much less expensive, so it drives the cost down for these products. Okay. You can also use insects, genetically modified insects. Insects are infected by baculoviruses. These are specific viruses that infect insects. And these viruses, before infection, are transformed. They're just a vehicle to insert mammalian DNA, and they cause the desired proteins to be produced in insect cells. Okay. Insects have different post-translational modifications, so that needs to be tested. And also, insect systems are usually small. It's very difficult to culture insect cells in a large scale, so usually this is small-scale production for experimental quantities of protein. Okay. Now we'll talk about downstream processing methods. Downstream processing methods are very important. They comprise the majority of the cost of producing recombinant proteins. And the first thing that you need to do is you have to harvest your extract. So once you have cells that produce the recombinant protein, you have to harvest the protein directly from the cells. In some cases, the protein is secreted, and so it's in the extracellular space, like the milk of goats. In other cases, the cells, like E. coli, have to be broken open so you can harvest the protein. Okay. The entire cell is harvested if the protein is intercellular, and so you have to lyse the cell to disrupt the cell wall and release the protein. And then, after the cells are ruptured, organic alcohols and salts are added to the mixture for stabilization. And if the protein is extracellular, then the culture medium is collected, and the culture medium is then purified. The second step, after you harvest the protein and you have an extract that contains the protein, you want to stabilize it. Okay? So this should be maintained at a lower temperature and at a proper pH that will prevent misfolding of the protein. You add protease inhibitors that will keep proteases from chewing up your protein product. And you add antimicrobials like antibiotics that will prevent the growth of bacteria in your product. You use additives to prevent foaming. Protein solutions like to make lots of foam. And you have additives that will prevent shearing of the protein. Next, you want to separate out the different components in the extract after the extract has been stabilized. And similarities between proteins allow proteins to be separated from other macromolecules like lipids, nucleic acids, carbohydrates. And then differences between the individual proteins can be used as targets for separation. If you have a protein that has a certain isoelectric point, if you have a protein that binds a certain substrate, all of these can be utilized to be able to purify out that single protein away from the rest of the proteins that are in the mixture. So quite often the proteins are precipitated out, and using hydrophilic amino acids on their surface, they attract and interact with water. Okay, this characteristic is used as a basis for separating proteins from other substances, and you simply add ammonium sulfate. Ammonium sulfate will cause the proteins to precipitate out, and you can also use other solvents like ethanol, isopropanol, acetone, diethyl ether, and they'll also, by increasing the affinity to the water, will cause the proteins to precipitate out. You can also separate out the proteins based on molecular size. This is a little bit more difficult. If your protein is a particular size, then you can use a membrane filter that has a specific pore size in order to get an initial purification on your proteins. You can centrifuge out the proteins. You can, based on your protein sedimentation characteristics, you can get the protein extract and get that away from other macromolecules. Proteins can be isolated in a single layer at the bottom of the centrifuge tube, or they can be separated from heavier cell components. Okay. And here's a centrifuge. This just shows a different tubes. The centrifuge is rotating. Usually you pull a vacuum. The vacuum allows the centrifuge to go faster. And you centrifuge, the cell homogenate, and afterwards then you have larger and dense materials, and then the supernate, which usually contains the protein, is in the lighter materials. Okay. You can also have a centrifuge that doesn't have these different tubes. You can just have what's called a batch centrifuge. And this batch centrifuge, you directly add the liquid through the separating discs. The protein comes out the top, and then all the cell debris flows out the bottom. Membrane filtration is used. Usually this is nylon or some type of other engineered polymer, and this can filter out the debris. This is faster than centrifugation. It removes any type of precipitates, any type of solids and bacteria. And then you can use a further method of purification called ultrafiltration, circle ultrafiltration and microfiltration. And this will catch molecules such as proteins and nucleic acids and separate out your specific protein of interest. Membrane filters are easy to clog. They're consumable, so they have to be replaced quite often. And so that makes them difficult to use and adds to the expense of downstream processing. Okay. You can also use dialysis, where you're looking at concentration differences of a salt buffer. This is used to remove small components, small contaminants like salts or stabilizers that you don't want in the final product. And you can use diafiltration as well, which is a type of dialysis. Dialysis is just when some molecules can't pass through the semi-permeable membrane and others can. This is used to remove smaller salts, solvents, and other additives. And then the salts are replaced with buffering agents that are usually safe for human use to stabilize proteins. And diafiltration is just a continuous process. It looks like dialysis, but you're continually replacing the salts that are used in purification with salts that are appropriate for use in the final product. Okay. So you start out with a dialysis bag, you seal it, okay, and you place the bag in a low salt buffer and you let it go to equilibration. The salts then flow out of the bag and the protein is retained in the bag. And then after several buffer exchanges, then the salt will leave the bag and then the proteins will be resuspended and purified. You can also use chromatography. And chromatography is a method that you can sort out proteins by size or how they bind or separate from other substances. A chromatography column is a long glass tube, and it's filled with resin beads and a buffer. The protein extract is then added, and it flows through the resin beads. And depending on the resin's property, the proteins will either stick to the beads or pass through the column while the beads act as a filtration system. Okay. And here's a chromatography column. This one's quite large. It's several inches in diameter, and you can see the protein solution, which is yellow, has been loaded and it's progressing through the chromatography. You can also see a lighter yellow where some of the smaller components go faster through the chromatography column, and those will be collected and discarded, so in favor of the protein fraction that's in the darker yellow. Here's an individual with a very large chromatography column. Usually these are done in clean rooms. The clean rooms have to be nearly sterile. They don't necessarily need to be completely sterile, but you want to prevent contamination by bacteria or any particulates. We'll talk about the different types of chromatography. The first type of chromatography is called size exclusion chromatography. And here you use gel beads that are porous, Larger proteins will work their way around the gel beads. They are too large to get into the pores. And smaller proteins will migrate slowly because they're able to enter the gel beads, and that retards their flow. Okay, so you have a chromatography column here. You add low molecular weight and high molecular weight substances. You let that flow through the column. The high molecular weight substances bypass the beads, and they elute first. And then the lower molecular weight protein fraction elutes later. It's held up in the column, and it's going to take a longer time to go through the column. So if you look at the progress of the chromatography column in time, high molecular weight proteins come out first, then intermediate, and then finally your low molecular weight fraction, and you just collect whichever fraction your protein product is in, depending on the molecular weight of the protein. You can also use what's called ion exchange chromatography, and this relies on the surface charge of the proteins that bind resin. Okay. The charged proteins will bind the resin, and the contaminants will pass through and out of the column. The proteins, if they're not eluted, they'll just stay in the column forever because of the electrostatic interaction. By increasing the ionic strength of the solution, you flood the column and all of its active sites with other ions. That releases the protein, and the protein then can be collected in the eluent. Okay. And here is a positively charged protein with negatively charged beads. I'm sorry, positively charged beads and negatively charged proteins. I got that wrong. Okay. And so the proteins, different fractions come through. Anything that doesn't bind the positively charged beads elutes. And then as you increase the ionic strength, then you get protein Y out, which weakly binds the charged beads. And then protein Z out comes out last, which strongly binds the beads. Okay, so what you do is first you just take buffer without salt and elute that through the column. The small molecular, non-charged particles come out first. Then the weakly negatively charged particles come out second as you increase the ionic strength. Okay, and then finally the highly negatively charged proteins come out last. And then you could, if this is your fraction, you can collect this fraction, and you've got a relatively pure fraction. The most specific type of chromatography is called affinity chromatography, and most proteins will bind specifically and reversibly to ligands. Ligands are usually small molecules, like substrates, that bind particular large portions of protein. And so you coat your beads with these affinity ligands, you put them in the column, and then you flow your protein mixture over the column. And after the proteins have bound to the resin, then you add a special buffer that will wash out anything that's not bound, and then add a final buffer that will break the bond between the ligand and the retained protein, and you can get the retained protein in the second elution fraction. Okay, here's a column resin bead, and it's got an affinity ligand that has a specific shape. Your protein product will bind this specifically. Okay, and then you have other proteins, contaminated proteins that you may have gotten out of the cell lysate. You're not interested in those. They'll elute out first because they're not going to bind the ligand, and your protein then will elute out second. You can also use what's called hydrophobic interaction chromatography. And these proteins will repel water, and they're sorted out on their basis of repulsion of water. The column beads are coated with a hydrophobic molecule like an oil, and then the hydrophobic amino acids like tyrosine in the protein will be attracted to the similar chemicals in the beads. It's just like attracts like. So something that's hydrophobic is going to attract to a hydrophobic bead. Okay. So here is your column resin. It has a hydrophobic affinity ligand on it. A protein that has hydrophobic portions will then break free from the bonds of water, and it will go directly and bind to the hydrophobic ligands. Okay? And then ultimately, what you do in this case is you start with an increasing salt concentration and then decrease the salt concentration over time. And the most hydrophobic proteins will elute last. They'll come out of the column last because they're tightly bound. Another method of chromatography is called isoelectric focusing. And you basically bind the proteins based on their electronic signature, their isoelectric point. This is where the point where the protein is electrically neutral. And the charges on the protein will match the pH of the solution. So when you have highly similar proteins that are similar from each other, and you want to separate them, then you separate them based on their isoelectric point. Finally, you have verification. So once you have a purified protein, you need to verify that the target protein was not lost. So you need to verify that when you run multiple chromatography steps, which is typical, usually you're running 2 or 3 chromatography columns. You want to make sure that your target protein is there, so you run out gels. You just do SDS page. And page is polyacrylamide gel electrophoresis, and you just use a detergent called SDS, sodium dodecyl sulfate, and that's added to the protein mixture and it's heated. The sulfate charges are evenly distributed from sodium dodecyl sulfate along the denatured protein, and that makes separation dependent on size. Okay, so you're not worried about charge, you're worried about size. So you can run these out easily in a gel based on size. Okay, and here's multiple purification steps. And you can see that the desired protein is augmented through step 1. You can barely see the band. Step 2, it's a little bit clearer. Step 3, the other bands are becoming fainter. Step 4, and then 5 is your control. Okay, so even after step 4 in your purification, you still have contaminants you have to deal with. You also want to verify the identity of the protein using Western blotting. When you have a Western blot, you use an antibody that's specific to that protein in order to probe the gel. So the proteins on the gel are transferred from SDS page to a nitrocellulose membrane. The sites on the membrane are blocked so the antibody doesn't bind non-specifically. The antibody is added. A secondary antibody for a detection is coupled with alkaline phosphatase, and that's added to the membrane. Excess secondary antibody is washed off, and then the substrate is added, and then that will cause a dye, a visible band, where the antibody is detecting your particular protein. Here is a protein that was expressed in plant tissues. So here you have the total leaf extract, the chloroplast extract. We're getting smaller. We're actually getting the thylakoid of the protoplast, or chloroplast, excuse me. And then if you extract photosystem 1, then you get your target protein. Okay, and this is done using page, SDS page, and then you blot it on a membrane, treat it with an antibody, and the target is what appears to be the darkest. Okay. You can also use ELISA if you don't want to run it out on a gel. You can just blot a membrane and use enzyme-linked immunosorbent assay. You need 2 antibodies. You need a detection antibody and then a reporter antibody. And you just use like a 96-well plate. Antibody 1 captures the unique protein, and then the protein is added to the plate where there's several watch. Antibody 2 sandwiches the protein, and it's attached to an enzyme covalently, and then the addition of the substrate allows a colored reaction if the antibody is bound to the protein. Okay. Here's an ELISA plate, and where you see a deep dark green, that is where the largest amount of detection is occurring. This will be read by a multi-plate or a multi-well plate reader, and then the wells that have the highest expression level will be used to verify the presence of the protein. Once the protein has been purified out, you need to preserve it. You need it to save it for long-term storage so the activity can be preserved. One way is to freeze dry the protein. That's called lyophilization. And lyophilization will maintain the protein structure over a long period of time. And freeze-dried proteins then can be stored at room temperature. Okay, then they can be reconstituted in some type of buffer solution. And I think I will stop at this particular slide, and this concludes the lecture that you're responsible for on Chapter 4 for Monday, February 6th.